Immunochemistry. Antigen. Any substance which evokes an immunological response is an antigen or immunogen. Epitopes. Antibody response will usually be selective against specific spatial configuration on antigen, which are called antigenic determinants known as epitopes. Immune response, cell mediated immunity, humoral response, effector mechanism, cell mediated immunity, immunity against infection, against bacteria, viruses, almost all parasites, rejection of allograft, body tries to reject implanted organs mainly by T cell mediated mechanism, tumor cell destruction. Helper function. T helper cells are necessary for optimal antibody production by plasma cells and cyclic T cells, cytotoxic T cells, Suppressor function. Production of lymphokines, delayed T type hypersensitivity. Naive helper T cells, here you can see interleukin 4 and interleukin 13 extracellular parasites antibody production by T cells, interleukin 17, interleukin 21 extracellular bacterial and fungal neutrophil activation, interleukin 10, interleukin 21 and interleukin for long-term humoral immunity and intracellular pathogen activation of macrophages. T cells are mobilized when they encounter a cell such as dendritic or B cells that has digested an antigen and is displaying antigen fragments bound to its MHC molecules. Cytokine cells the T cell mature. The MHC antigen complex activates the T cell receptor and the T cell secretes cytokines. Some T cells become helper cells and secrete some cytokines that attracts fresh macrophages, neutrophils, other lymphocytes, and other cytokines to direct the recruits once they arrive on the scene. Infected cells. Some T cells become cytotoxic T cells and attack down cells infected with viruses. Some cytokines spur the growth of more T cells. Humoral immunity. Antibodies are produced by plasma cells, and one common thing is that, like here you can see, for humoral immunity, antibodies are released, and for cyto T cells are released. Okay. The antigen antibody reactions leads to activation of complement system which destroys the foreign cells. The antibodies can destroy the large cells by following mechanism. Classical complement pathway, antibody dependent cell mediated cytotoxicity, agglutination and opsonization of target cells. AB cells is triggered when it encounters its matching antigen. The B cells engulfs the antigen and digests it. Then it displays the antigen fragments bound to its unique MHC molecules. The combination of antigen and MHC attracts the help of mature matching T cells. Cytokines secreted by the T cell helps the B cells to multiply and mature into antibody producing plasma cells. Released into the blood, antibodies lock onto matching antigens. The antigen antibody complexes are then cleared by complement cascade or by the liver and spleen. Stem cell, early pro-B cells, late pro-B cells, large pre-B cells, small pre-B cell and mature B cell. CD 34 cluster determining or differentiating, okay, CD19, CD40, CD43, CD45, class 2 MHC, major histocompatibility complex cells, NEB cells, GCB cells, plasmablast short-lived, memory BB cells, bone marrow malt, mucosal associated lymphoid tissue, spleen red pulp, IgG, IgA, IgM, IgG, and IgA. Spleen red pulp is having long-lived plasma cells IgM, IgG, IgA, and malt IgA and bone marrow IgG. Antibody dependent cell mediated cytolysis. ADDC requires antibody, but the cytolysis does not require complement activity. The effector cells are neither T nor B cells but are called K or killer cells. It is effector at sites of tumors. Structure of immunoglobulins. The Ig is made up of two heavy chains and two light chains combined through disulfide bridges. Depending on the heavy chain makeup, the Ig are differentiated into five major classes: IgG, gamma heavy chains, IgM, mu heavy chains, IgA, alpha heavy chains, IgD, delta heavy chains, and IgE, epsilon heavy chains. So you can learn it by game firstly and then just add a D. IgG, IgA, IgM, IgE, and IgD. Okay. The light chains are either kappa or lambda in all the classes. In human beings, 60% K type light chain, 40% lambda type light chain. Variable and constant regions. Both heavy and light chains contain relatively variable V and constant C regions with regard to their amino acid composition. VL and CL, general terms for light chains. VH and VC for heavy chains. Variable light chain, constant light chain. Variable heavy chains, variable constant chain. The first 108 amino acids in light chain and first 118 amino acids in heavy chains constitute the variable region. The amino acid sequence can vary in heavy and light chains so that the body could synthesize enormous varieties of different antibodies. So this is the structure of an antibody. Here you can see antigen binding site and light chain and heavy chain. Like as you know, birds are light weighted, so you can learn that this light chain is like bird which is flying, okay, in the air. So this is the fact, like light chain are in the air. Okay, light chain and heavy chain, VL and CL and VH and CH, CH and VH, CL and VL. Light chain, hinge region, complement activation, heavy chain, cell binding, heavy chain, antigen binding, heavy chains. The immunoglobulin molecule NH2 amino terminal and C double bond OH carboxy terminal and constant region are shown as dark. Variable heavy chain, variable heavy region, variable light chain, constant heavy region, Cl constant light region chains are connected by disulfide bridges shown as SS linkages. Fab FAB and FC portions, papain proteolytic enzyme from papaya cleaves the Ig immunoglobulins so that two FAB fraction antibody portions and one FC fraction crystalline lysable portion are produced. The antigen binding part of antibody is in Fab segment. Another proteolytic enzyme papain cleaves Ig at another site so as to yield Fab AB2, where two Fab portions are combined. Fab AB part can combine with antigen very weakly, but combination with FAB is stronger. Different classes here you can see IgG, IgA, IgM, IgD and IgE. Nomenclature of Fabicians: gamma, alpha, mu, delta, epsilon have its chain domains 4, 4, 5, 4, 5. Number of basic 4 peptide units 2 light and 2 heavy. IgG, IgA2, IgM5, IgD1 and IgE1. Additional unit S and J and JP's molecular weight Daltons 1,46,000, 3,85,000, 9,70,000, 1,85,000, 1,90,000. Sedimentation coefficient 7s, 11s, 19s, 7s and 8s. Carbohydrate content 2 to 3, 8 to 10, 12, 10 to 13, 11 to 12. Concentration in normal serum per 100 ml 800 to 1200 milligrams, 150 to 300 milligrams, 50 to 200 milligrams, 1 to 10 milligrams, 1.5 to 4.5 microgram. Half-life in days 26, 10, 3, 2. Distribution intravascular 45%, 5%, 95%, 75%, 50%. Functions of immunoglobulins. Placental transfer, as you know, IgG can cross the placenta. Complement fixation, IgG, IgA and IgM is having the most of the complement fixation function. Agglutination, IgM and IgA and IgG. Binding to macrophage, IgG. Fixation to mast cell, IgE. Primary response antibody is IgM. External secretions, IgA. Natural antibodies, IgM. And here is the structure: IgG, IgE, IgD and IgM is pentamer, and IgE is like two combines. Here you can see IgG, IgE and IgD have one basic unit each. IgM has 5 basic units and IgA has 2 basic units. Red circles represent J pieces, green squares are secretory pieces. Complement system. Complement factors are proteins present in serum. Cell lysis by antibody is mediated by complement system. They are abbreviated as C1 to C9. The complement activity is abolished if serum is incubated at 56 degrees Celsius for 30 minutes, as most of the components are thermolabile. The half-life of the complement system at room temperature is only few hours. Classical pathway: antibody fixing on antigen on cell surface. Alternate pathway: microbial polysaccharides, endotoxin, immunocomplexes, etc. Prop, properdine, factor D, factor B. Characteristics of complement system. Complement component: electrophoretic mobility. Main site of synthesis is epithelium of intestine, and here you can see reticuloendothelial cells, hepatocytes, reticuloendothelial cells, macrophages, hepatocytes, macrophages and macrophages. Primary and secondary immune responses. When an antigen is injured, antibodies start appear in 10 days, reach peak level in 20 days, response decline by about 30 days. IgM will be predominant in the primary response. When the same antigen is injected into the same animal after a few months, the response is quick, within 3 days. Secondary response is, here you can see, is quick. Secondary or anamnestic response is quick, is stronger, more avid, IgG type, more prolonged for months. Secondary immune response. Okay. Active immunity induced by immunization with toxoid or killed or attenuated organisms. DPT vaccines, polio vaccines. Passive immunity protection is given by perform preformed antibodies. This is used in immunotherapy against snake bite or tetanus toxoid, Clostridium tetani. Okay, passive immunity preformed antibodies are provided. Okay, so here you can see the graph like secondary antibody response, repeat infection, cell plasma, and amount of antibodies, long-lived plasma cells in bone marrow, memory B cells, and primary antibody response, first infection, short-lived plasma cells in lymphoid organs, activated B cells, IgG, IgM, low-level antibody production, long-lived plasma cells in bone marrow. So here you can see antibody concentration and the time period, first injection of antibodies, lag phase, declining phase in primary response, and their secondary response. Here you can see the peak is like here you can see, and this one is for primary response 1 degree, and this one is secondary response. Soluble factors: interleukin 1 alpha and beta, secreted by macrophages and other antigens presenting cells, induces interleukin 2 receptors, induces acute phage proteins, co-stimulation of APCs and T cells. Interleukin 2 activated T helper cells, maturation of T and natural killer cells into LAK, lymphokine activated killer cells. Interleukin 4 T helper Th2 cells, B cells proliferation, IgE expression on B cells. Interleukin 6 activated Th2 cells, hepatocytes and adipocytes, acute phage response, B cells proliferation synergistic with interleukin 1 and TNF on T cells. Interleukin 8 macrophages, chemoattractants for neutrophils and T cells. Interleukin 10 T suppressor CD8 cells inhibit cytokine production, suppresses cell immunity. Interleukin 15 mononuclear phagocytes induces production of natural killer cells. Interleukin 18 macrophages increases natural killer cells activity, induces production of IFN gamma, IFN alpha, macrophages, neutrophils, production of macrophages and NK cells, antiviral effects. IFN beta, fibroblast, antiviral effects. IFN gamma, activated Th1 and NK cells, differentiate cell lines, antiviral effects, activates macrophages, neutrophils, and natural killer cells. C-reactive protein (CRP), hepatocytes, adipocytes. Label of C-reactive protein parallels inflammatory responses in the body. Level is increased by interleukin 6. CRP enhances phagocytosis by macrophages, induces expression of various cell adhesion molecules. ICM VCAM selectins attenuates NO production by NO nitric oxide production by down-regulating NOS expression. TNF alpha macrophages inflammation, fibrosis, pyrexia, acute phage proteins necrosis. TNF beta T cells inflammation. GCSF macrophages stem cells stimulation of granulocytes. GM-CSF T cells and macrophages, stem cell stimulation of granulocytes and macrophages. MIF T cells, activation and inhibition of mobility of macrophages. IL for interleukins, IFN for interferon, TNF for tumor necrosis factor, GCSF granulocytes colony stimulating factor, GMCSF granulocyte macrophages colony stimulating factor, MIF macrophage migration inhibition factor. Thank you. Go and download med notes. It is very helpful.